And so I'm just going to jump into this by expressing this idea of one of the interests we have in the lab is how the immune system and the brain relate. So common features with what the brain does and what the immune system does. So something that's super cool about our brains is that we can learn, we can remember things, and integrate new information and have that influence future decisions about our future actions and behaviors. And so the immune system does this as well. It's kind of like a brain that's floating around in our bodies that's able to integrate new information, recognize environmental stimuli, and sort of undergo a learning process, and store memory about this and have that influence future behaviors just like the brain does. And so I think it's just such a cool thing that we're interested in the lab. And even though it seems like the immune system might be less sophisticated than the brain, there are things we could do with the immune system kind of memory that we can't yet do with the brain. For instance, we can't track a specific memory in the brain yet, like a neurological memory, but we can do that with the immune system, which is super cool. Like for instance, if you remember something that happened when you, as a child or something, you can't really physically know where that is, or you can't track the magnitude of that, or it's using a biological assay. But with the immune system, you can, and that's super cool. So this interest of ours in terms of how the immune system remembers stuff and learns is relevant to the pandemic. And so in our lab, we develop cohorts and try to learn stuff about the immune system in the context of the SARS-CoV-2 pandemic. I think it's fair to say that current problems we face now include evolving SARS-CoV-2 variants and waning immunity. And the immune solutions to that are broad recognition and protection, as well as long-term memory and long-term commitment to this kind of protective response. And so what we do in the lab is we study antibodies. I'm a B cell biologist, and my lab studies B cells and antibody production evolution. And so just to make just to kind of briefly do this, to kind of summarize this background, I'll just say that memory in the antibody system can be summed up in this slide by just showing that there are memory B cells which express antibodies that have some specificity that the immune system has seen before, and that these memory B cells, they're cells, and so they can respond to this again, and they can continue to evolve and adapt and so forth, and change the antibody affinity and mature further with this antibody. But the other component of memory, another component of memory in the antibody system, is secreted antibody. So these are plasma cells, that developed from other B lineage cells, like memory cells can turn into plasma cells, that secrete their antibody. And so these are terminally differentiated plasma cells, and they can live a long time or not, but they tend to secrete the antibody, and the antibody is floating around, as proteins in the body. So that it's kind of a backup system, but at the same time, I don't think these are completely redundant. So, secreted antibodies can perform certain functions that a memory B cell may not be able to do all the way, and just in terms of timing of the antibody availability and how it can protect and so forth. So some redundancy, but at the same time, they offer some protection that is not redundant. So how are antibodies produced? There's really three ways. This shows two ways of the three the antibodies are produced. The way that's not shown is natural antibody production. That's just, independent of really any stimuli. We produce kind of anticipatory antibodies that float around in the body. There are other two ways the antibodies are produced. These are activation induced. So the B cell sees some inflammatory stimulus and then can respond to that by making antibodies. One way is a quick way extrafollicular pathway. So this is where B cells can quickly differentiate into antibody secreting cells as well as memory cells through the extrafollicular pathway. You can get class switch recombinations, so you can go to initially produced IgM to IgG. And in general, I think it's fair to say that we think that this produces more short-lived plasma cells. Then there's the germinal center pathway, where B cells undergo sort of a more sophisticated developmental program, where there's more coordinated interactions with more cell types in an anatomically, configured way. You have other cell types like follicular dendritic cells and specialized T cells and so forth. And here, B cells can also develop into plasma cells to secrete antibody, as well as memory cells. I think it's fair to say that there's a couple of unique features with this pathway, and that is that here B cells tend to be able to undergo more somatic mutation and affinity maturation. So antibodies can evolve in real time, receive mutations in the antibody genes themselves, and then evolve with these mutations to enhance their affinity from good usually to even better. And I think it's also fair to say that that we think that long-lived plasma cells tend to come from a germinal center dependent pathway as well. So that's the special thing about about the germinal center pathway, and that's highlighted in yellow. There's somatic mutation, long-lived plasma cell. Okay, so to get into what we were doing to look at, just to look at some aspects of these questions, we have 3 cohorts that I'll tell you about. So here they're represented in these colors: green, purple, and gray. So green cohort had mild COVID and they recovered, and we followed their antibodies over time, both memory B cells as well as the antibodies secreted in the plasma, and before they got the vaccination. So this is the green, had mild COVID, not yet vaccinated cohort. Then there's a purple cohort. These folks had COVID, most of it was mild, and then received the vaccination. We continue to follow them. And then there's a bunch of people that we followed that had not had any evidence of COVID before, and they got vaccinated, and we followed the immune response to that as well. And so we measured, how antibodies recognize the virus, in particular the spike protein of the virus, the strength and durability of the antibody response longitudinally over time, and how vaccination versus natural infection looked. And so, as I mentioned, we were interested in the spike protein of the virus. So the spike protein is one of the major antibody targets, and antibodies that target the spike protein can be neutralizing, meaning that they can prevent the virus from getting in the cell in the first place. And so this is a schematic of the protein structure of the spike protein. So it's a trimer, and so shown on the left is a protomer where you have the 2 main parts of the spike protein shown, S1 and S2. S2 is shown here in red. It's more conserved between coronavirus family members. The S1 part is kind of broken up into the N-terminal domain, shown in purple, middle image, and the receptor binding domain, which is in red. Receptor binding domain is this little domain that can flip upwards, and that's the configuration where it kind of interacts with the host cell. Okay, so one of the things we did was we looked at how people recognize the spike protein. What parts of the spike protein are recognized more in folks compared to other parts of the spike protein, and how does that correlate with any functional activity of the antibody? Does where it binds, does that influence its function? And so here is a bunch of antibodies we cloned from the green cohort here. So this is had COVID, not yet vaccinated. And we cloned a bunch of antibodies here, shown 109 antibodies. and we just wanted to clone them in an as unbiased way as possible. So we basically just sorted memory B cells that were spike reactive, and then we just took all comers, spike reactive B cells, and just asked, where do they bind? So we cloned the antibodies from the cells, and then to kind of get at this, we just did this big cross competition experiment where we took all the antibodies and just asked, do they compete with each other? And so shown here, this is all the antibodies we had on the top, or at least 109 antibodies that worked for this assay are on the top. And then listed on the side are the same antibodies. And we just ask, okay, does this one compete with this one? And we just went down the list. And so when we did that, a pattern emerged where there were 6 major competition groups, 7 total that we can really identify with confidence, that kind of blocked out in these sort of blocks. So that the darker orange means that they competed more strongly. So there were 3 main ones in the RBD, and there were 2 main NTD ones, and then there were 3 S2 domain ones. And we were able to then map these out with other known antibodies, as well as mapped out the function of these antibodies. And we found that, the RBD2 was the major super neutralizing target. RBD1 and 3 had some potential to do this, but NTD1 also was a very super neutralizer. S2-1 was the domain that was able to be targeted by antibodies that recognize broadly across, a bunch of other coronaviruses, like, the common cold coronaviruses. And when we took all the, from these 19 individuals and just asked, where people recognize spike, this pattern emerged here. That's just shown on the right. We took a couple of individuals out of the total just to see that there is some heterogeneity, like this person G32 mostly recognized RBD2, the super neutralizing piece, and this person C12 targeted more RBD1. But in general, this is sort of the hierarchy of targeting in people. And basically they're named based on the targeting frequency. So RBD1 was targeted more than RBD2 and so forth. All right, so what about the variants of concern? So, we're worried about the Delta variant now. It's super contagious. It's the one that's going around mostly. There are other variants that have come out in the past. These are just schematics that show where the mutations are that we won't go over right now. But in terms of these domains that we mapped here, we just named here, they're listed here on the bottom. In blue, this is like where the antibodies can still bind to the variant spikes. And so you see here that, for beta and gamma, RBD2 was not targeted really well anymore. If the antibodies were induced by the parent, they sort of lost binding to some of these mutants. But Delta, for RBD2, you still get a bunch of binding. NTD1 is also one of those that tends to escape antibody recognition. And again, RBD2 and NTD1, these are where you can find some super neutralizers. Okay, so the panel on the bottom, this is neutralization activity. So the darker purple means better, more potent neutralization. So I think just the thing to point out here is that despite the fact that you get a bunch of escape from some of these variants, there are antibodies that are made by people that continue to recognize and potently neutralize all of the variants. So, we do make a small frequency, at least, of broadly, at least in terms of variant, SARS-CoV-2 variant, broadly neutralizing antibodies when induced from a parent strain. So there is that within the human repertoire. So this is just a schematic we published with that paper recently. I don't think I go over it. We went over pretty much all this stuff. It's just, the frequency of targeting and their functional properties and their ability to continue to recognize variants. All right, so what about strength and durability? So here we're showing the purple and the gray, in addition to the green, antibodies over time. So green had COVID, recovered. Here are their antibodies, days post-symptom onset, out past 200 days. And then the purple, people that got, recovered from COVID, got the vaccine. This is how they looked over here in terms of after first dose is up here. And then after the second dose, days after that. So both purple and gray, people that did not have COVID and got the vaccine. And so a couple things to point out. One is that natural infection tends to be, the antibodies induced by natural infection tend to be modest, at least in terms of having mild COVID. They're modest antibodies, but they tend to hang on. There's some decay, but they hang on pretty stably over time. Whereas if you compare that to folks like myself, I haven't had COVID, so I'm in this, gray group, but got vaccinated. So robust amount of antibodies induced after the second dose, but the antibodies do tend to sort of decay over time. And more antibodies quicker in the purple group, and the decay does tend to be more gradual in the purple group as well. All right, and then let's see, this is just plotting out the data points per day. So without the binning of the, of the previous slideshow, just to show that the slopes. There is less of a decline in terms of the slope for the purple, and green is the stablest, whereas black tends to decrease, where by 200 days, we're at the level of natural infection-induced antibodies. All right, so what about the variants? And so here is just showing the original Wuhan strain, the D614G mutation up on the top, and then alpha, beta, gamma, delta, looking at sort of the first and the last draw we had for the green cohort. And then, this green shading pretty much shows the amount of neutralization capability that folks with natural infection had by 200 days, and that's kind of shaded here in green, just as a comparison. And you can see that folks that just got the vaccine, and in particular with, if you look at the Delta variant, that immunity, at least in terms of neutralizing antibodies, the neutralizing antibody kind of immunity decreases down where a bunch of people fall below where we might want to be by this time point. That said, there are still neutralization, measured. There's still neutralization measured by this point, but how that's going to sort of plateau, I think time will tell. All right, so this is to point out that antibody magnitude matters in terms of just at least neutralization ability when we're thinking about the variants. So last year we published a paper where we parsed out the green cohort. We basically asked, okay, among the folks that have had COVID and recovered, what's the heterogeneity look like in terms of antibody durability? And there's a subgroup of people that we called sustainers, and what this means is just that they continue to make as much or more antibodies later as they did at the beginning. There's a subgroup of these sustainers that continue to make antibodies, even out to like past 200 days. They make as much, if not more, compared to about a month after they had their symptom onset. So this is after they recovered, measured their antibodies, and they continue to make about the same amount. And so when we looked at all the others, which we call Decayers for lack of a better term, these folks tend to just sort of get less, although they do plateau and in general maintain. But when we look at, if we parse these people out, sustainers in red, Decayers in black dots, and we look at the different variants, we see that sustainers have superior neutralization function towards all the variants. And that's pretty much, a function of just having more antibodies. So as I mentioned before, we all make a small frequency of antibodies that are broadly neutralizing. And so if you make more antibodies, you'll have more of that small frequency, and that holds up across the variants. And so you can see that here, these donut plots. The sustainers and decayers, about half of them were cross-variant neutralizing capabilities, whereas over time, the sustainers maintained their neutralization capabilities, whereas the decayers lost that ability. Okay, so we wanted to look into this a little further in terms of the sustainers. Why potentially, what makes a sustainer? So this is a schematic that just highlights what a sustainer is. They had shorter symptoms, they had sustained antibodies. They also, their memory cells had higher early somatic mutation. And so this graph here kind of shows that. It's bimodal peak that we found, the really high somatic mutation, anti-spike memory B cells, and kind of a lower one here. When we look at the decayers, they basically had, almost no mutation in their memory B cells early on. That said, they both, over time, like 3 months, there's a substantial increase of somatic mutation in everybody. And so we are interested in looking at this further. So we cloned memory B cells from the sustainers and decayers, and we divided them up actually as well. So the super high ones, mutations, as well as the lower ones. I just wanted to point out that both of these modes had higher mutations compared to the companion modes on the decayer side. When we looked at this and we plotted the memory B cell antibody clones, here are the different colors, just indicate different people. We ranked the antibodies from highest somatic mutation to lowest. Here in purple, you can see that. And then when we looked, all of the super high mutated ones, they all cross-reacted with other coronaviruses, in particular, the beta coronaviruses. So SARS, MERS, HK1, OC43, NL63-229E, they're more distantly related coronaviruses. We didn't see cross-reactivity with these, but we did see a bunch of cross-reactivity with the other coronaviruses, and these antibodies also tended to cross-react with the S2 part of the spike protein, which is the more conserved part. So, so we thought that was kind of interesting. So, perhaps this is immune recall from prior infection or something, and that, or they just were able to hang on to these memory cells longer for whatever reason, because, and this might have some antibody durability influence. And then the other sort of summary point, magnitude matters, especially, when we're in light of the variants.
Duane, sorry to interrupt. This is the 2-minute warning.
Oh, thank you. All right, so in the last 2 minutes, I'll just mention this thing about what about antibody breadth? So here we're talking about magnitude in conferring breadth just because you have more of stuff of a composition where you have some low frequency of broad binders. What about on a per antibody basis? So we are interested in this question as well. So we took this neutralization data that we had across variants, and we wanted to ask on a per antibody basis, what about the composition of the repertoire of antibodies induced by either natural infection or vaccination? So we took the neutralization potency to the variant and divided it by the original strain, which our infection cohort was before the variants had emerged. And so, we think that the variants, or at least that the kind of the spike in the vaccine is similar to what, the folks were infected with in our cohort. So the long story short is that the green and red here, this is the natural infection. The green is natural infection 200 days out, induced antibodies. And the purple and the gray, if you compare, the gray does have less breadth on a per antibody basis. If you just look here at the delta, less breadth compared to the natural infected induced antibodies, less breadth compared to also the purple group, which had vaccine plus the natural infected induced antibodies. So, and there's some interesting patterns here, but that the take-home message is that antibodies induced by natural infection may have a slightly superior breadth on a per antibody basis. So just to kind of summarize here, the breadth can be a modulatable thing depending on the immune stimulus. As well, we could talk later if there are questions about, immune recall point that I mentioned. Memory B cells increase in somatic mutation over time. If you have a higher affinity, that might influence the long-lived plasma cell piece of it. And so I mentioned all of these. And so just to end, the main point here is additional aspects of immunity might be within reach for sort of continued improvement of vaccination strategies. So I needed to point out here, Yuezhou Chan, Peitong, Adam Zuyani, and Avnish, who are the main folks behind the work. So thanks so much for your time.
Thank you, Duane. And I'll just remind attendees and panelists, please raise your hands and we'll call on you. I saw there's a question posted in the chat, but if we can get a hand up, that would be great. And I'll kick us off, Duane. I have a question about the durability of the antibody response for the 3 different cohorts. It looked like the vaccinated group had the most rapid decay in durability out to day 200, but in a sense, they were all plateauing at a similar level unless that decay continues out past the 200-day mark. And I'm just wondering, have you followed these folks past that 200-day mark?
So yeah, this is brand fresh new data from our cohort. We're kind of up to where they are in giving their blood. And so that's a great question. And yes, we're following them out. We don't have the data yet just because they haven't come in yet for their blood draws after that long. But I would say, if they do plateau where natural infection had given, for instance, I think that may be sufficient maybe for the natural virus, the original virus, but for variants that might be, that's where it gets kind of a problematic, because with variants like the Delta variant, it protects some, but not everyone, you know what I mean? So
yeah.
Whether it plateaus or not, it's still kind of a concern.
So I guess the big question is, are we going to need boosters?
Yeah, I think boosters will certainly be interesting to see. Is that going to be enough recall, effect, and to stabilize the slope also, in addition to do the magnitude boost, which would help as well.
Wayne, could I ask, what do we know about J&J or adeno versus mRNA?
Yeah, great question. Folks on the call might know more than me on that. We only have 7 people in our cohort with J&J. And we are now, running those because they ended up coming later than the mRNA folks did in our cohort. So we actually, in our cohort, we don't know yet out past 6 months, but I'm sure J&J does, and other folks that might have that data.
So almost all your data is mRNA anyway.
Correct. Yeah, everything that I showed here is mRNA because those are the folks that had completed the 200 days so far. We only have 6 or 7 that have J&J that's completed, maybe 4 or 5 months by now.
Right. Gotcha. Thanks.
And if I could ask one more question, can you comment on what is the difference between a sustainer and a decayer? Does it have something to do with the viral load upon initial infection, or we just don't know yet?
So we defined the sustainer only based on the antibody levels at a later time point compared to the beginning blood draw, which was usually, a week or 2 after symptoms resolution, and then, about a month after symptom onset. The initial antibody magnitude for both sustainer and decayer was very similar. So I imagine viral load might be related to antibody response. And so we didn't measure the antibody, the viral load. I wish we had that data, we don't. So it could be that the viral load might have been different, but that said, that doesn't explain why their antibody levels initially were similar. The sustainers did recover quicker, and which, so they had less duration of disease. And then they also had memory B cells that had higher somatic mutation. but that was after we defined them based on their durability statistic.
Okay, thank you. We have
At that point, could I ask, is there any data for serious symptoms for the people who are really seriously infected as opposed to mild?
Yeah, so we have about 5 of those in our cohort, and they tend to have super high antibody levels early after, and they decline down to sort of the average, by 200 days. But that's all we know. So the more serious symptoms they had, the higher early antibody levels they had. Yeah.
Okay, and we have a question from David Cohen in the audience. And I realize I do not have host function. So if Jesse or Esther could unmute David.
Can you hear me?
Yes.
So my question is, do you have any data on the response of these cohorts to any other vaccinations that they may have received during their lifetimes?
So we don't yet, but we're super interested to follow up on their flu vaccine response. Because most of them are health workers, or a lot of them are healthcare workers, and they would, they're getting that every year.